Hey everyone! In the last video you learned how mitosis works. Now in this video we'll take a look at what can go wrong during the cell cycle, and we'll also examine the cell cycle checkpoints that are designed to detect and respond to these occurrences. As always, remember to fill out the learning guide, and by the end of the video you should be able to answer 2 questions. And our 2 questions are: why is cell cycle regulation important for growth and development? And what errors in the cell cycle does each cell cycle checkpoint detect? Cell division in multicellular organisms is not uncoordinated. Cells don't just divide continuously. This would result in a blob of cells. We can actually produce these blobs in cell culture. In plants, these are called calyces. Cell division is coordinated or regulated in that the timing of cell division of all cells in an organism takes place according to a set plan that is ultimately determined by the organism's genes. This requires communication between cells through hormones and growth factors. This regulation is what allows cell division to produce an organism with well-defined tissues and organs. In fact, we know enough about the regulation of cell division that if we supply the right pattern of growth factors and hormones, we can actually get this blob of cells to become a plant. We can even do this to some extent for human tissues and organs. So, what is the regulation of cell division? Essentially, it's the control over whether a cell divides or completes the cell cycle. Cells divide only at the appropriate time and place, and only if certain conditions are met. Some cells may be actively dividing, while others are not. This can be nicely visualized by looking at the growth of plant roots. The highest rate of cell division in the root occurs at the tip, in this area. Once the cells are made, they get longer or elongate. The production of cells and their elongation is what makes the root get longer and longer.
This coordinated division of some cells but not others
occurs because the division of these cells are upregulated, while the division of these cells is downregulated. The plant upregulates cell division at the tip, so these cells divide faster. Cell cycle checkpoints, which we'll discuss later in this video, is one way this regulation is accomplished. At this point, go ahead and view a time-lapse video of this process by clicking the first link below this video. In the video, the flashes of green indicate when a cell is dividing, so you'll see most of the flashes at the root tip. I'll give you a few seconds to check it out. So far, we've discussed why regulation is important for growth. Regulation of the cell cycle and cell division is also essential for the replacement of dead cells and for the prevention of uncontrolled cell division or cancer. Let's consider these 3 functions of cell division regulation in the very hand that you're using to operate the computer or device that you're watching this video on. Now, your hand was formed when you were a fetus in your mother's womb. The fingers and hands form more quickly, and the thumb forms more slowly. So at some point, the cell division of the cells in your fingers had to be downregulated, while cells in your thumb had to be upregulated. This type of regulation allows the orderly construction of complex structures and organs in multicellular organisms. Regulation of cell division is also important for cell replacement. This is important for healing. Perhaps you've had a cut on that hand of yours. When tissue is damaged, cells surrounding the damaged site are induced to divide to help heal the wound. Follow the second link below for a time-lapse video of a wound healing, and think about how the cells dividing at the wound site help to heal it. I'll give you a few seconds to check it out. This is possible because cell division is upregulated at the site of the wound. Regulation of the cell cycle is also important for preventing the buildup of mutations, which can lead to unregulated cell division and cancer. Regulation of the cell cycle helps prevent cells with harmful mutations from dividing, preventing the mutation from being passed on to other cells. The regulation of cell division is accomplished through the enforcement of cell cycle checkpoints. Next, we'll look at several things that can go wrong during the cell cycle. We'll look at the effects of these errors, and we'll look at the checkpoints that detect these errors. Here is the cell cycle that you've seen in previous videos. 3 of these checkpoints are the G1S checkpoint, the G2M checkpoint, and the spindle assembly checkpoint. These are transition points when the cell determines whether to proceed through the cell cycle, or to pause the cell cycle, or in some cases, destroy itself outright. Now, during G1, the cell grows. A cell might fail to grow because it lacks growth factors or nutrients. If such a cell divides and keeps dividing, the cells will get progressively smaller, which might lead to abnormal development. The G1S checkpoint checks that the cell is large enough and has the right nutrients and growth factors to divide. The G1S checkpoint is the main decision point that determines whether or not a cell will divide and proceed through the rest of the cell cycle. It occurs during G1 right before S phase. During S phase, the chromosomes are replicated. But if not enough DNA replication enzymes are produced during G1, the replication of the chromosomes might not be completed, and some parts of chromosomes might not be replicated. The incompletely replicated chromosome would be missing many genes, which could lead to cancer or other genetic diseases. To help avoid this, the G1S checkpoint also checks if enough DNA replication enzymes have been produced during G1. During the cell cycle, damage to the DNA of chromosomes may occur. Many environmental factors, such as excessive exposure to sunlight, radiation, and mutagenic compounds or carcinogens, can damage or mutate DNA. But DNA damage can also occur naturally. 2 harmful types of DNA damage are double-stranded breaks and thymine dimers. Double-stranded breaks are when one chromosome breaks into 2. Thymine dimers are when new bonds are introduced between 2 Ts that are next to each other on the same strand of DNA. both types of damage can destroy the function of the gene that they occur within. If these mutations are in genes that regulate cell division, and if there are enough of them, it can lead to cancer. For example, the p53 gene, which is needed for pausing the cell cycle so the cell can repair damaged DNA, is often mutated in melanoma, which is a dangerous form of skin cancer. Additionally, the mutations are often thymine dimers, which makes sense given that this type of mutation is caused by UV radiation from sunlight, and melanoma is known to be caused by excessive exposure to the UV radiation in sunlight. The G2M checkpoint is in G2 and right before M phase, and checks for DNA damage. The G1S checkpoint also checks for DNA damage. If DNA damage is detected, the cell pauses the cell cycle, allowing DNA repair enzymes to repair the damage. Or, if that's not possible, the cell may destroy itself through apoptosis or programmed cell death. The cell actually checks for DNA damage throughout G1, S phase, and G2, but it can halt the cell cycle at either the G1S or G2M checkpoints if DNA damage is found. Since the G2M checkpoint is after S phase, it also checks that all chromosomes have been completely replicated. Another thing that can go wrong during the cell cycle, specifically during mitosis, is the uneven distribution of chromosomes. Here is a cell in metaphase of mitosis. Normally, each of the two sister chromatids is attached to a spindle fiber from opposite ends of the cell. However, if one of the two sister chromatids is not bound by a spindle fiber, both chromatids of that chromosome may be pulled to one side of the cell, resulting in one daughter cell with 3 of that chromosome type, and one daughter cell with only 1 chromosome of that type. Failure of two sister chromatids to separate during anaphase is called non-disjunction. If we examine the word non-disjunction, disjunction means to separate, so non-disjunction means the sister chromatids do not separate. This is in contrast to disjunction, which is occurring normally for all these other chromosomes. These two cells are aneuploid. In other words, they have an abnormal number of one chromosome type. So non-disjunction is one cause of aneuploidy. A cell with only one of a chromosome, like this one, is called monosomic, while a cell with 3 of a chromosome type is called trisomic. Aneuploidy can arise during mitosis, as shown here, or during meiosis, and can lead to genetic diseases and syndromes. We'll examine aneuploidy and its consequences in more detail in a later video. The checkpoint that prevents non-disjunction is the spindle assembly checkpoint. Even though I've drawn a mitotic metaphase cell here, this checkpoint actually acts during prometaphase, because this is when the spindle fibers are attached to the kinetochores of sister chromatids. So the spindle assembly checkpoint holds the cell in prometaphase until both sister chromatids of each chromosome are attached to spindle fibers from opposite ends of the cell. This is good because separation and sorting of chromosomes into daughter cells is irreversible. You can't take one chromosome from one cell and put it back into another. How exactly does the cell determine if both sister chromatids are bound to spindle fibers from opposite ends of the cell? We'll discuss this and the mechanisms of the other checkpoints in the next video. So, we've discussed the G1S, G2M, and spindle assembly checkpoints. To review, the G1S checkpoint checks for DNA damage, cell size, growth factors, and nutrients. The G2M checkpoint checks for DNA damage as well and ensures that the chromosomes have been completely replicated. The spindle assembly checkpoint checks that each sister chromatid is bound to a spindle fiber from opposite ends of the cell. There are also other cell cycle checkpoints, but we'll end our discussion here. Okay, so at this point you should understand why cell cycle regulation is important for growth and development and understand the function of each of the checkpoints that we discussed. If you don't, feel free to watch part or all of the video again and make sure to complete the learning guide. Now, in the next video, we'll take a look at how each of these checkpoints work. See you then.